Hi all, it's Dr. DT. This is the first of two videos that will go along with the histology outline. In this one, we're going to look at some general information and then we're going to go through the epithelial tissues. In the second video, we'll go through the connective tissues, muscle tissue, and nerve tissue. Histology can be described as the microscopic study of tissues. So we're going to look at things that are pretty teeny tiny, and we're going to need a little piece of equipment to help us do that. And we're going to do a lot of that in the lab. If we were in class, I would ask you to define the term tissue, because we've had that already. And what we would say is that these are two or more cells with similar structure that perform a common function. Hopefully you remember the principle of complementarity of structure and function. And that reminds us that structure and function are highly correlated. So we'll see when we look at these tissues that what they look like and how they work go together really nicely. And this might be one of the better examples of complementarity of structure and function. Hopefully you also remember that there were four basic types of tissues that are integrated to form the human body. So we are going to have epithelium or epithelial tissue. We're going to have connective tissue. We're going to have muscle tissue. And we're going to have nervous tissue. And so we're going to go through each of those different kinds of tissue. We're going to do them in that order, and we're going to see what they look like and how they work. And then finally, we know that tissues are going to combine to form the next layer of hierarchical structure, and that would be organs. So most organs are going to contain all four basic tissue types, and we'll see that as we start to study them. This picture from our book reminds us of the jobs of the four different tissue types. So nervous tissue here is going to be involved in communication. It's going to form brain, spinal cord, nerves, that kind of stuff. Muscle tissue, we know, is going to be involved in movement. Epithelial tissue is going to form boundaries, and then it also will form glands. We'll talk more about that. And then finally, connective tissue is going to be sort of a supportive tissue. And again, we'll talk about examples of that. We're going to start off with epithelial tissue, and I really love epithelial tissue because it's really well organized and it makes a lot of sense once you get how it's put together. So epithe here as a prefix literally translates as covering. And so when we look at the functions of epithelial tissues, they're going to serve as either coverings or linings for the body's external and internal surfaces. So they're going to form a boundary between different areas of the body. And that's going to have a number of different important functions. One thing that we'll do is provide some protection. We will be able to do absorption and secretion. Epithelial tissue will be involved in filtration, excretion, and finally, sensory reception. So we'll see these functions, reception. Let me try that again. There we go. So we'll see examples of those functions as we go through, both 282 and 281 and 282. All right, the second function for epithelial tissue is to form glandular tissue. So we're going to start by going through the coverings and linings, and when we're done with that, we're going to look a little bit at glands. So this is a picture that I stole off the internet, and I really like it because it shows you the different parts in sort of a simplistic view. I would draw it, except I'm going to be using the same picture over and over and over again, and I thought instead of drawing it, we'll just use this one. So I'm going to tell you that these cells that we're seeing right here, I'm going to highlight one of them in blue. That's going to be part of my epithelium. And there'll be several other cells that are like it. And then underneath it, we're going to have some connective tissue. I'm going to abbreviate connective tissue CT. And then you can see there's kind of a membrane looking thing in between them. We're going to talk about all of these parts. So if I look at the general structure of epithelium, what I'm going to find is it's kind of a sheet of closely packed cells with very little extracellular space in between them. And so we say that we have a characteristic that's called cellularity. So very cellular tissue here. So again, if I look, here's a cell, here's a cell, here's another cell, and so on, and there's very little space in between those cells. Now, we might even see things like tight junctions in between the cells to prevent material from moving in between them. We may also see desmosomes that are connecting the cells, holding them again tight together. The next characteristic of epithelium that we're going to see is something called polarity. So polarity implies that we have sidedness. There are two poles, so sort of like the North Pole and the South Pole, or if you're looking at chemistry, a positive end and a negative end. And so here what we're going to have are going to be an apical and a basal surface. So let's start off with what's called the apical surface, and that is the free surface of the cell, or in this case of the tissue, that is exposed to the external environment or an internal cavity. So I'm going to tell you that this area here that I'm going to kind of color in yellow may be the external environment. It may be the internal body cavity. And we're going to give that area a fancy name. We're going to call it the lumen. So the lumen here is the external environment or the internal cavity, depending on what we're talking about. So for example, if we're looking at the skin, imagine that this is the surface of your skin here. So this is, would be the fat that you have under your skin and the muscle that you have under your skin. And then this area would be the lumen. So this would be the area that's outside your body. If we were looking at a tube, for example, like the intestine, in the middle it's going to be hollow, and that middle part, again, we would call the lumen. And oftentimes there's something in the lumen. The epithelial tissue are going to be the cells that are right next to the lumen here. Okay, so on this picture, the apical surface is going to be the surface that is right next to the lumen. The basal surface then is going to be sort of the bottom surface or the deep surface or edge of the cell or tissue that is attached to the rest of the organ. So on this picture, the end down here is going to be our basal surface. So all within the epithelium, the top is the apical surface, the bottom is the basal surface. Now, sometimes we're going to have epithelial tissue that is more than one cell thick. So imagine that we had several cell layers. So here I'm going to make several cell layers. Okay. None of them's going to look really like this, but just imagine that we have this. And here will be that membrane underneath, and then the connective tissue will be underneath that. So the apical surface will be here, again next to the lumen, and the basal surface will be down there next to that membrane. So we're going to look at the cells that are next to the lumen when we're identifying the type of epithelium, so keep that in mind. All right, the next characteristic that we want to remember about epithelial tissue is that it is avascular. So A, we know, means without, and vascular here means blood vessels. So there are no blood vessels in epithelium. And so it turns out that, if you remember, these are living cells, so they're going to have some kind of support tissue underneath them to make sure that they stay alive. And the support tissue that's going to do that is going to be the connective tissue. So let's take a look at that. Epithelial tissue is going to be supported by underlying connective tissue, and that stuff has a name. We're going to refer to it as the lamina propria. Sometimes I'll just abbreviate it LP for lamina propria, not liquid propane. And so down here, this connective tissue, again, LP for lamina propria. So the lamina propria is going to be separated from the epithelial tissue above it by this membrane that we can see right here. And it has a double layer here. And I'm going to label that BM, not for bowel movement, but for basement membrane. Okay, so basement membrane, BM. And that is a protein matrix that separates the epithelium from the connective tissue above it. So it turns out that the basement membrane has two layers. We're going to have a basal lamina and we're going to have a reticular lamina. The basal lamina is the one that is right next to the basal surface of the cells. So it's the top one, it's the one that's closer to the epithelium. The reticular lamina is going to be the sort of deeper or bottom layer, and it is closer to the connective tissue. They are made out of slightly different materials. The basal lamina is made out of glycoproteins and collagen. The bottom layer is going to be made out of just collagen. I'm not super worried about us remembering that. Functions of the basement membrane are going to include being sort of an epithelial boundary. So again, separating the epithelium from the underlying connective tissue. It can help act as a filter. It will provide some scaffolding or something to build structures upon. And then it is also going to help us resist stretching and tearing. And that was a very loud car that just went by. The next characteristic we want to remember about epithelial tissue is that it is innervated. And basically this term, if you look at it, it looks like it has nerve, the word nerve in it. And basically it means that the tissue is supplied with nerve fibers. So it is going to be sensitive. And the last characteristic is a high regenerative capacity. So regeneration means that we can sort of make it over. And in this case, it means that the tissue can do rapid mitosis. And if you think about it, that's pretty important because this tissue is subject usually to a lot of abrasion and possible damage. And so if it is damaged, we can repair it pretty quickly. Epithelial tissue is going to be classified on the basis of two characteristics. The first one is the cell shape, and the second is the cell arrangement. So let's start by looking at the cell shape. There are three that are talked about in your book, and then there's one additional one that we're going to mention. The first is called squamous. So think of squamous. Squame here, this term means like shingle. So if you think about like the shingle that's on your roof, these guys are flat. So they're thin, they're flat, they're maybe kind of scale-like if you look at them. And so this would be sort of what you would see if you looked at the squamous cell from on top. And this is what they'd be if you looked at them from on side. So again, these are thin, flat. Sometimes from on top they look like a fried egg. From the side, they're very thin. The second shape is cuboidal. So again, we have that cube in the word. I like to think of these as kind of square, at least when I'm looking at them again from the side. If I look at them up from on top, they don't look quite so square. But if I was going to draw this, I would sort of maybe draw it like this, and that will be our cuboidal cell. Squamous cells are going to have their width be greater than their height. In cuboidal cells, the width and the height are going to be the same. Columnar sounds like column, and so I think of these as maybe sort of rectangle shaped. Apparently I can't spell rectangle, but here we're going to find that the height is greater than the width. So if I look at the picture of the columnar over here, again, definitely taller than they are wide. If I sort of sketch this one out like this, it looks sort of like a rectangle. Transitional cells are going to change their shape. So they are not always the same. And so sometimes we'll find them looking kind of like a rounded edged square, sort of like this. And then they can go back and forth between that and being sort of flat. So depending on what they're doing, they will change shape. We'll talk more about that. There are two types of arrangements talked about in your book, but we're going to add one additional arrangement to our group here. The first arrangement is simple. Here we're going to have one layer of cells, and you can see it over here in the picture. So if I look, I'm going to have a basement membrane. So I'll have a basal layer, I'll have a reticular layer here. I'm going to write this as BM for basement membrane. And then if I look, here I have one layer of cells. Stratified will have two or more layers of cells here. And so again, if we look for the basement membrane, which I'm going to draw in here, there's our two layers of basement membrane. Here would be the lumen. And by the way, over here, I'll put the lumen here as 2. Anyway, here if I look, I'm going to have, looks like 1, 2, 3, 4, 5 layers of cells. That's definitely more than 2. That would be stratified. So pseudostratified is kind of a weird thing. It looks like it's stratified, but pseudo here means false or fake. So there's really only one layer, but it looks like there are more. If you were able to get a really good look at pseudostratified epithelium, you would be able to see that all the cells actually touch the basement membrane, but sometimes it's hard to see that because the cells tend to be at different heights. All right, finally, if we put the shape and the arrangement together, we can use that to name epithelial tissue types. For example, we have simple cuboidal, we have stratified squamous, and so on. So we're going to look at the different types and see how this works. For each type of epithelium, we're going to look at first a cartoon. We're also going to look at a real picture from underneath the microscope. We're going to talk about the structure, the function, and some examples. So we're going to start with simple squamous epithelium. So simple here tells us that we should have one layer, and squamous tells us that we should have flat cells. So one layer of flat cells is what we should see with simple squamous. So let's look at the picture over here. So here in purple, we are going to have our basement membrane. They tried to make this look three-dimensional, but the lumen will be up here. So in between the basement membrane and the lumen, looks like one layer of cells, and if I look at those cells, they look flat. So again, tight together, not very much space in between them. The function of simple squamous epithelium is going to be to allow the tissue to do either rapid filtration or rapid diffusion. So we're going to have thin cells so that materials can get through them really quickly from one side to the other. Examples of places where we might find this would include the lung, and that's what the picture that we're going to see on the bottom here is. And then parts of the kidney. There's a blood vessel in there that's called the glomerulus. And there are other places too. You don't have to know glomerulus for right now, but kidney is another place where we're going to find simple squamous epithelium. So let's look at the real picture here, because this is a little harder to see, and this is what you're going to look at underneath the microscope. So this is going to be from the lung, and if I look at the picture, this is a lumen, this white space. Here's another lumen, here's another lumen, 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 so on. Okay, so all the white spaces are lumen. So the tissue right next to the lumen here is going to be our simple squamous epithelium. Now, it's really hard to see a basement membrane in there, and these cells are really, really thin because we're actually seeing 2 layers of them in there. So if I took this and I blew it up big time, it would look sort of like this. So I'm going to have a layer of thin flat cells that's right next to the white space. I'm going to have a basement membrane with the two layers, and then I'm going to have another layer of thin flat cells on the other side. And again, there'll be a nucleus in there, so it'll look sort of like that, but again, super thin. All right, now it turns out that there are also two kind of lookalikes of simple squamous epithelium that are called endothelium and mesothelium. And sometimes people say that they are simple squamous epithelium, but I'm here to tell you that they are not, because they come from a different embryological derivation. So let's take a look at those two tissues. The first one I have here is endothelium, and what I might want to remember about this stuff is that it's going to line blood vessels, it's going to line lymph vessels, and also the heart. And so if I look at these cells here, I can see them again right next to, this is the lumen here in a vein, this is the lumen here in an artery. And the cells that are right next to the lumen here and here and here, those are going to be endothelial cells. They're actually derived from something that's called endoderm embryologically. We don't need to remember that, but they have a little different shape. So from the side, they kind of look like elongated triangles that make up a sheet that is the membrane there. They are super slick, and they are going to help decrease friction in blood vessels, lymph vessels, and the heart so that the blood flows really smoothly. So this over here in the picture is actually, looks like maybe a capillary there, and we're going to see that same endothelial cells here and here and here. So again, you can see how very thin these guys are. The other look-alike tissue is called mesothelium or mesothelium, and this is what makes the serous membranes in your ventral body cavity. So if we were in class right now, I'd ask you what those serous membranes are. There are three of them. You should know them. There's one around the heart, one around the lungs, and one around the abdominal pelvic cavity. So this is sort of what the one around the peritoneal cavity looks like from on top. So if you're looking top down, you'll see these kind of big cells that look kind of like fried eggs, kind of like what simple squamous epithelium looks like from the top. Here we're looking at it kind of from the side. So this would be the lumen. And so the cells that are right next to the lumen are going to be our mesothelium cells. And again, they are flat little cells. This is not a perfect view through it. We're seeing it at kind of an angle because really this should just be one cell layer thick. Our next type of tissue is simple cuboidal. So again, simple reminds us that we're going to have one layer of cells. Cuboidal is going to tell us that they are kind of square in shape. And so if I look over here at the picture, again, I can see the basement membrane here. The lumen is going to be here. And here I have one layer of cells between the lumen and the basement membrane, and they definitely look like this, they have the same height and width. Now, one other thing that I might notice when I look at these guys is they are going to have a big, round, centrally located nucleus. And I'm even going to put that in my description, okay? So square with a big, round, centrally located nuclei. All right, so that's going to be important when I look at the real picture over here because sometimes it's hard to see the plasma membranes. So first thing I'm going to do is I'm going to find the lumen. So here's a lumen, here's a lumen, here's a lumen, 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 so on, okay? Then this is my basement membrane. Here's another basement membrane. Here's another basement membrane. Here's another basement membrane, and so on. So I may not see the plasma membrane, but I'm going to see big, round, centrally located nuclei. And if I sort of imagine where the membranes would go, Now I have cells that I can see look pretty square. So again, that's an important thing to be able to do. All right, what's the job of these cells? Well, they have more cytoplasm. So because they have more cytoplasm, I got more space. I can put more organelles in there. And so these guys will make stuff and secrete it. So they'll need to bring things in. So secretion and absorption are big functions of simple cuboidal epithelium. We're going to find this stuff in glands. We will also find it in our kidneys, in a thing that's called the tubules of the kidneys. But again, I don't need to know that right now. I just want to remember I can find some of this in my kidneys. In lab, we're going to look at the thyroid, which is an example of a gland, and that's where we'll see our simple cuboidal epithelium. The next type of tissue is going to be simple columnar. So again, simple tells us we're going to have one layer of cells. The cells are going to be taller than they are wide. So one layer with, I don't know, let's put rectangular cells. We're going to notice when we look at these cells that we have kind of an oval nucleus, and that oval nucleus is going to be at the basal end of the cell, so the bottom of the cell. So if I look over here at my picture, again, here's that basement membrane, here's the lumen. We're going to have one layer of cells. The cells are definitely taller than they are wide, and I have kind of an oval nucleus that's towards the bottom. And if I look, I'm going to see that my nuclei all line up pretty nicely in a row. And that's one of the things that we might be able to see in simple columnar epithelium. Now, simple columnar epithelium may have two other specializations here. One of the specializations is microvilli. We already know about microvilli, and if we were in class, I'd ask you what they do. And so you would then say these guys are going to increase surface area. So they're going to be little brush border kind of extensions of the plasma membrane up there at the top. The other thing that we sometimes will see are things called goblet cells, and they tend to stick in here in between adjacent cells, and I always think they look kind of teardrop shaped and maybe kind of frothy when you look at them. So goblet cells are going to secrete mucus, and that mucus is going to serve as either lubrication or some kind of protection from abrasion. So these cells are also going to be involved in absorption and secretion, and you might imagine that we'd be able to do it even better because we have even more cytoplasm than we do when we're looking at simple cuboidal. So let's look at the picture from the microscope. This is from the stomach, and here would be our lumen, and we are going to find actually two areas where we have basement membrane. So we're having basement membrane here, and then we have another layer of basement membrane down here. So again, between the lumen and the basement membrane, one layer of cells. These nuclei look kind of round. They should be a little bit maybe more oval, but you get what you get, right? So one layer, tall cells, nuclei towards the basal end of the cell. So again, if I'm thinking about examples of where I can find this, I would say my GI tract. GI stands for gastrointestinal tract. So I'm going to find this stuff in my stomach and in my large and small intestines, except for the anus. Our next example is pseudostratified columnar. So columnar, again, tells us that we're going to have rectangular sort of cells, so they're going to be taller than they are wide. Pseudostratify here tells me that I'm going to have that false stratification. So I'm going to have one layer of cells that looks like multiple. And the reason that it looks like multiple is that these rectangular cells are going to have nuclei at different levels. And some of the cells are probably going to be shorter than others, but all of them will rest on the basement membrane. So let's look at the cartoon picture. Here's my basement membrane. Here's my lumen. So if I look, here's a cell, rests on the basement membrane, basement membrane, basement membrane, basement membrane, and so on. Okay? If I look at it from far away though, because the nuclei are sort of all over the place here, that looks like they're really stratified, but they're not. All right, so this is sometimes hard to see when you're looking at it underneath the microscope. So let's look at the real picture here. So down here is the basement membrane. So this tissue is twisted a little bit. Here's the lumen right there. And so if I look, I'm going to have a cell here, and all of these cells are supposed to touch the bottom. Some of them will be shorter, some of them will be taller, some of them it's not going to look like we hit the bottom. The nuclei are sort of all over the place and messed up. So again, this is a pseudo-stratified. It's messy though, sometimes it's hard to tell. Now this thing that I'm seeing right here is something that we talked about in the other picture, but we didn't point it out because it didn't have one. And that would be a goblet cell. So again, kind of a teardrop thing, looks a little frothy. We're making some mucus. That mucus will be put out into the lumen. So I might want to add to my structure here that I might see goblet cells. And I might also see these structures right here, which you might remember are cilia. And the job of cilia is to move materials that are outside the cell. So these guys are going to wave, and as they do, we're going to move stuff that's out there in the lumen. The function of pseudostratified columnar epithelium is going to be secretion and absorption, plus we are also going to secrete mucus. Oops. And finally, we're going to move particles or sweep away particles that are in the lumen. So nice examples of places where we'll find pseudostratified columnar epithelium are going to be in parts of your respiratory tract. So not the entire respiratory tract, but something that's called your trachea, which is also known as your windpipe. We'll find this in the larynx, which is also known as your voice box, and then some of the tubes that are going down into your lungs, like the bronchi. Our next type of tissue is called stratified squamous. So here now we have a different word. Stratified means that we're going to have 2 or more layers. Squamous, again, tells us that these guys are going to be flat cells. So let's look at the cartoon. Here is the basement membrane. Here is the lumen. So definitely between the lumen and the basement membrane, we have lots of cell layers. So that part makes sense. But if I look at the shape of those cells, the cells up here look to be pretty flat. The cells down here look to be cuboidal. So that's a little confusing. But remember, the cells that we want to look at are going to be the ones that are near the lumen. So we don't care so much about the ones that are near the basement membrane. Those may be cuboidal. They may actually be columnar. But these are the ones that we're going to look at to see the shape that we're going to use for the name of those cells. The picture that we have that's from under the microscope, the basement membrane kind of goes like this here in this picture. This would be the lumen. So again, we can see multiple layers of cells, and the ones up here at the top are going to be kind of flat. So let's think a little bit about structure function here. Before we had simple squamous epithelium, very thin tissues, and we used it for rapid filtration and diffusion. So we didn't have very thick cells. It was very easy to get stuff from one side of the cell to the other. Here we have a whole bunch of cells, and the function here is going to be protection. Remember, this tissue is going to form coverings and linings, and sometimes we need to have protection. And it might be protection from abrasion, it might be protection from dehydration, chemicals, all kinds of different things. So if I look at places where I'm going to find stratified squamous epithelium, one really obvious place is your skin, your mouth, your throat, your esophagus, your anus, and if you have one, your vagina. So these are all places that are subject to abrasion. We need to have a nice protective tissue there to make sure that the tissues don't rip. Our next example is stratified cuboidal. So again, stratified, 2 or more layers of cells. And these guys are going to be square, again, with those big round nuclei. And so if I look at the picture, here's the basement membrane, here's the lumen, and sorry about the quality of this picture, your book didn't have one. But if I look here, it looks like I have 2 layers of cells, and again, they're kind of roundy or kind of square here. So again, stratified cuboidal epithelium. Their job is going to again be secretion and absorption, just like I had with the simple cuboidal, plus we're going to do some protection, okay? So because I have multiple cell layers, protection is going to be important. So I want to look down here at the professional picture. So here will be the lumen. You can't really see that. Let me try writing that again. The lumen, and this one happens to have an egg in it because this is from an ovarian follicle. Down here, I'm going to have the basement membrane, and it actually goes all the way around this structure. And then if I look, I have multiple layers of cells. I can't see the plasma membrane, but I'm going to have big round nuclei that are relatively equally spaced, and that reminds me that they are cuboidal. So if I drew in sort of what this plasma membrane would look like, if I split the difference between the cytoplasm equally, might look sort of like that. All right, so where do I find these? These guys are rather insignificant, there we go, in the body. In other words, we don't have a lot of it in our body. We'll find it in a bunch of places, but not very much of it. So where do you find it? Well, we're going to see it in the ovarian follicles, which is that thing over there. We might also find a little bit of this stratified cuboidal in the ducts of our sweat glands. We might find a little bit in the ducts of the mammary glands, salivary glands. Okay, so again, not super important. We are going to again look at the ovarian follicle in lab. We won't look at the rest of the areas. The next tissue is called stratified columnar. So again, stratified, 2 or more layers. Columnar tells us these are kind of rectangular or tall cells. And so if I look over here at the picture, this is another one off the internet because your book didn't have a picture. Here is the basement membrane. Here is the lumen. I'm going to have multiple layers of cells, and the ones at the top here, the apical cells, are going to be rectangular. I'm going to look at the microscopic picture next. The basement membrane is probably this area right here. Here would be the lumen. This one looks like it only has 2 cell layers thick, but again, the top cells are taller than they are wide. The main function of our stratified columnar is going to be protection, but we're not going to have a whole bunch of this stuff either. So this stuff is relatively insignificant in the body. And so we'll find it in a bunch of places, but again, not very much of it in any of these places. So the places would include things like the male urethra, small areas of your pharynx or your throat, part of your epiglottis, anus, mammary glands, salivary glands. So kind of all over again, but not very much of it anywhere. This stuff sometimes is hard to tell from pseudostratified columnar. And so I'm going to make a little note here with a big star. And I'm going to tell you that we have no slide of stratified columnar for lab. So no slide in lab. So on a lab test, if you're looking at something and you're like, is it pseudostratified or is it stratified columnar? The answer is pseudostratified. So do not put stratified columnar as an answer on your lab exam. So every semester I say this, and every single semester I have at least one person who writes stratified columnar as an answer for the lab exam. And I already know it's going to be wrong because we don't have a slide of that. All right, the last type we have is transitional. So transitional here tells us that we are going to change shape. The cells are not always going to have the same shape. So this will be multi-layered, even though the multi-layered part is not in the name. The cells are going to change shape depending on whether the tissue is sort of relaxed or distended and full. So sometimes it has kind of a, kind of a balloon shape. And by balloon shape, I mean kind of a rounded square. So we don't want to confuse it with stratified cuboidal, and the cells that are below it will look a little different, so you shouldn't, but don't. And then sometimes it's going to look flat, and it'll look sort of like this, okay? So it will be balloon shaped when the structures are empty, and it'll be flat when the structures are full. What structures are we talking about here? Basically, we're talking about structures from the urinary system. So the picture that we have here is actually from the bladder. And so if I look, here's the lumen. Here's going to be sort of my basement membrane here. So multi-layered of cells. And the cells at the top kind of have that balloon shape, or kind of sort of rounded, squared, weird shape to it. All right. We are always going to see the cells in their balloon shape. And here's why. If you have a full bladder and you cut it to make microscope slides, all of the pee is going to be released, and then it's empty, and then they go back to their balloon shape. So sometimes, we're going to find this stuff in your ureters and your bladder and your urethra. And so sometimes this stuff is also called urothelium because it's only found in the urinary system. The function here is basically, basically to stretch. And so it's going to stretch in response to pressure, and that will give it more room so we can accommodate more urine volume. Oh, the cat's here. Can you hear him? Hi, Tom. Go away. I have to finish up glands now. So the last kind of epithelium thing that we want to talk about are the glands. And so glands are going to be one or more cells. So sometimes they're only a single cell. Sometimes there's a bunch of cells that work together, and they are going to make and secrete some kind of product. So it turns out glands come in two different flavors. We're going to have glands that we call endocrine glands, and then we're going to have glands that we call exocrine. So endocrine and exocrine are our two types. So I always like to do this by maybe drawing a little bit of a picture here. And so we're going to do multicellular glands here. And if I started off here, you might not be able to tell the difference between an endocrine and an exocrine gland until I add this structure to my exocrine gland. And that is going to be a duct, D-U-C-T, not D-U-C-K. So a duct is going to be like a little tube or a little straw or something, and it is going to take products that are made by the exocrine gland cells, and it's going to move them out of the exocrine gland cells to either the external body surface, so we're going to go either to the skin or to some kind of internal body cavity. So maybe that would be the inside of your stomach. Endocrine glands are ductless. In other words, they don't have any ducts. So they're still going to have a product, and the product is going to be secreted from the cells. It's going to go into the interstitial fluid, and then it's going to go into this red tube. And if you are being especially astute, you will recognize this, of course, as a blood vessel. So the exocrine gland products are going to go into a blood vessel, and then they can travel around the body. Wherever the blood goes, they can go as well. The last difference that we want to talk about between these two is what the products actually are. So endocrine gland products are going to be hormones. And there are lots of different hormones, and we're going to talk more about those a little bit in this class, but also a lot in Bio 282. The hormones are going to be regulatory chemicals. The exocrine glands are going to make a whole variety of different products, depending on what gland they are. And these variety of products may include sweat, oil, saliva, bile, digestive juices, mucus, and milk, and some other stuff, but that's probably a good enough variety. This is a picture from your book that looks at different kinds of exocrine glands. And so you can see they come in both a simple and a compound duct structure. And there are tubular secretory structures and alveolar secretory structures. And you know what? I don't care about any of those. So do not waste your time memorizing those out of your book. So that brings us to the end of our epithelial tissues. In the next video, we're going to look again at the connective tissues, muscle tissue, and nervous tissue.